For most of us, the phrase Persian blood carries a kind of comfort. It suggests a single unbroken thread, running backward through centuries, all the way to Cyrus and Darius, untouched by everything that happened in between. It's a clean story. It's also not what the DNA says. In 2025, a team publishing in Scientific Reports set out to test that assumption directly. They pulled 50 skeletons from 9 archaeological sites scattered across Iran, spanning nearly 6,000 years of continuous human presence. The earliest remains dating back to 4700 BCE, the latest reaching forward to 1300 CE. Inside that single dataset sits almost the entire arc of what people mean when they say Persian history. From those 50 individuals, researchers newly sequenced 23 mitogenomes and 13 full nuclear genomes. This wasn't easy material to work with. Some of the coverage on these ancient genomes was thin, as low as 0.20x in places, meaning the scientists weren't reading a clear signal so much as straining to catch a whisper preserved in old bone, fragment by fragment, base pair by base pair. And what that whisper revealed doesn't match the simple version of the story at all. The headline finding is continuity. Genuine, measurable continuity across nearly 3 millennia. But not the kind most people would guess. Not a pure bloodline preserved by isolation. Something stranger and more complicated, sitting underneath 4 different empires. What if the DNA said the throne changed hands over and over again? Dynasty after dynasty, conquest after conquest. But the people living underneath that throne barely moved at all. That question is the spine of everything that follows. If you're the type who likes finding out the empire you learned about in school wasn't the whole story, hit subscribe. There's a lot more of this. Most people carry an unspoken assumption about ancient Persia, that the Achaemenid Empire, the Parthian Empire, and the Sassanid Empire each represent a genetic changing of the guard. New rulers, the thinking goes, new blood. As if political conquest rewrites a population the way a new government rewrites a flag. The 2025 study says otherwise. Across those imperial transitions, the majority of the ancient Iranian gene pool remains stable. Not stagnant, not sealed off, stable. At least half of the ancestry found in medieval southwestern Iranian samples traces directly back to Neolithic Iranian farmers, populations that were tilling soil in that region thousands of years before any of these empires existed. That signal didn't survive by accident. It passed through Iron Age populations first, carried forward generation after generation, before it ever showed up in the medieval samples researchers eventually pulled from the ground. 3 separate eras, one continuous thread of ancestry running underneath all of them. What makes this genuinely surprising is the context surrounding it. This wasn't a region tucked away from the world. It sat in constant contact with Mesopotamia, with the Levant, with the Caucasus, with Anatolia, a crossroads absorbing merchants, armies, and migrants for centuries on end. And it still didn't get genetically overwritten. The evidence for all of this is thinner than you'd expect, historically speaking. Only 2 medieval genomes currently exist from the entire Iranian plateau, a striking gap given how thoroughly documented this region is in written history. One of those 2 genomes came directly from this study. To understand why that kind of stability is surprising, you have to go back further, back to what the plateau actually was before any empire ever rose on top of it. Genetically speaking, the Persian plateau isn't a country. It's a vast geographic zone, stretching from eastern Anatolia all the way to Afghanistan and western Pakistan, sweeping in the South Caucasus and reaching into northern Mesopotamia along the way. Long before it hosted any empire, this stretch of land served as a corridor for early Homo sapiens leaving Africa, somewhere between 70,000 and 46,000 years ago. Thousands of years later, trade routes traced almost the same paths those early humans once walked. Silk Road branches threaded south of the Caspian Sea, cutting straight through the same corridor, carrying goods, ideas, and people back and forth for centuries. The researchers behind the 2025 study gave this specific stretch a name, a genetic highway. Their work marks the first genetic evaluation ever conducted on this particular corridor, a section of the ancient world that shaped migration patterns for tens of thousands of years, but had never actually been tested at the genetic level until now. That's part of why the 9 sites in this study weren't chosen at random. Each one sits directly along this route, positioned to catch whatever genetic traffic passed through over the millennia. And that's exactly what makes the underlying finding strange. A region engineered for movement, built, quite literally, as a highway for migration, somehow produced one of the more stable, continuous gene pools in all of West Asia. Stability on a highway sounds like a contradiction. The deep prehistory of this place is where that contradiction starts to make sense. 8,205 years before the Common Era. That's the date attached to the lead sample in one of the foundational papers on this region. Bruschacki and colleagues, published in the journal Science in July of 2016. Their team sequenced 4 early Neolithic genomes recovered from the Zagros Mountains, with that lead individual dated to a window between 8205 and 7756 BCE. What they found reshaped assumptions about early farming populations across the entire region. These Zagros farmers were not ancestral to Europe's first agricultural populations. They barely registered in the ancestry of modern Europeans at all. Genetically, they had already split away from Anatolian farming groups an estimated 46,000 to 77,000 years earlier, an enormous span of separation for 2 populations that would go on to build agriculture in roughly the same era, just a few hundred miles apart. Their genetic fingerprint didn't vanish, though. It shows up today in specific modern populations. Pakistani and Afghan groups carry some of that signal, but the single strongest affinity of all belongs to one very particular community: modern-day Iranian Zoroastrians, whose genetic profile lines up with these 8,000-year-old farmers more closely than almost any other population researchers have tested. One of these ancient skeletons even carried a Y chromosome marker researchers can trace by name. Her name in the archaeological record is simply Ganj Dareh, the site where she was found, in what's now western Iran. Researchers sequenced her genome at 1.39x coverage, and she lived roughly 10,000 years before present, at a settlement with some of the earliest evidence anywhere for an economy built around goat herding. Genetically, she sits closest to Caucasus hunter-gatherer populations, a different lineage entirely from the Anatolian farmers who would later carry agriculture into Europe. Her community, like the Zagros farmers found later, made little direct contribution to the ancestry of modern Europeans. The Zagros individual researchers labeled AH2 carried Y haplogroup J2BDM12, a lineage still traceable through the region's genetic phylogeny today, thousands of years after his death. Researchers describe Western Iran during this period as somewhat isolated from the rest of the Fertile Crescent. A population the 2025 study's earliest new sample doesn't come from a Bronze Age palace or an Iron Age fortress. It comes from the early Chalcolithic period, and it predates every other Chalcolithic genome ever recovered from Iran. Before this discovery, researchers simply didn't have a data point from this exact window. This individual fills a gap that had sat empty in the record. What's inside that genome matters as much as its age. The ancestry is mostly early Neolithic Iranian, the same Zagros farmer signal traced back through Bruschacki's science paper and the Ganj Dareh genome, still present, still detectable, thousands of years after those original farming communities lived. Alongside that core signal, researchers also found evidence of some Western genetic influence beginning to enter the mix, the first sign of outside contact layering onto the older Zagros base. Researchers framed this sample as proof of long-term cultural and biological continuity, centered specifically on the Zagros area. Not a population wiped clean and replaced. A population absorbing new genetic material while keeping its older foundation largely intact. Functionally, this single sample does something no other piece of evidence in the dataset does. It bridges nearly 8,000 years of Neolithic genomes on one side to the historical period samples, Achaemenid, Parthian, Sassanid, sitting thousands of years later on the other side of the timeline. It's the single oldest data point directly connecting the deep prehistoric past to the Empire era at the center of this story. Move the clock forward roughly 2,000 years from this Chalcolithic sample, and an entirely different force starts reshaping the region. Not drifting in from the west this time, but pressing down from the north. Narasimhan and colleagues, publishing in Science in 2019, generated genome-wide data from 362 newly analyzed ancient individuals, bringing the full study's total to 523 people. Tucked inside that number is something researchers had never had before. The first ancient genomes ever recovered from eastern Iran and from Turan, the region covering modern Uzbekistan, Turkmenistan, and Tajikistan. What that data revealed was a clear southward push of steppe ancestry into Turan during the Middle Bronze Age, somewhere between 2300 and 1500 BCE. This wasn't a local shift. The genetic profile of this steppe signal matches Bronze Age Eastern Europe almost exactly. The same population movement, branching outward toward 2 very different destinations on the map. Researchers connect this migration to something beyond genetics entirely: the shared linguistic features running between the Indo-Iranian language family and the Balto-Slavic languages spoken thousands of miles away. One population, moving in multiple directions, apparently carrying both genes and grammar along with it. The scale of that impact shows up clearly in modern populations. In parts of South Asia today, this same steppe ancestry accounts for up to 30% of total genetic makeup, a substantial fraction, given how far south and how long ago this movement began. Eastern Iran sat directly in the path of that migration, and one site there, positioned right along the same corridor, produced genomes that simply didn't match the people living around them. Shari Sokhta sits in eastern Iran, and during the Bronze Age, it functioned as a major trade hub positioned directly along this same migratory corridor. Excavations at the site turned up something researchers didn't expect from a typical local population: outlier individuals whose ancestry profile lines up with populations connected to the Indus Valley civilization, hundreds of miles to the southeast. These outliers stand apart genetically from the general population buried at the same site. They weren't locals with slightly different genetics. Their ancestry profile points somewhere else entirely, toward the urban centers of the Indus Valley rather than the surrounding Iranian communities. That connection matters beyond this one site. Shari Sokhta represents one of the earliest solid genetic links researchers have found between the Iranian plateau and the urban civilizations of South Asia. Physical evidence that people, not just goods, moved along this trade route. Burial customs from this same broader region tell a parallel story. Catacomb-style burials, a tradition associated with the Persian plateau, appear at related sites after roughly 2700 BCE, later than a similar tradition found at Jiroft further south. The oldest catacomb graves anywhere on the plateau predate the ones near Shari Sokhta by centuries, placing Jiroft's version of this practice well ahead of its eastern counterpart. That burial style has its own genetic story, 300 kilometers away and 3 centuries earlier. Jiroft's catacomb-style graves date to between 3000 and 2900 BCE, the oldest known examples of this burial style anywhere on the entire Persian plateau. Centuries later, both Herodotus and Strabo documented catacomb burial customs among Parthian tribes, describing a practice that, by their time, had clearly outlived its original context by a very long margin. The style itself appears to have spread outward from southeastern Iran, influencing burial practices as far as Baluchistan and southern Turkmenistan, a single funerary tradition, radiating outward from one region, absorbed by neighboring populations over time. Then the tradition simply disappears. For more than 2,000 years, catacomb burials vanish from the archaeological record on the plateau. There's no clear evidence of the practice continuing during that long gap. It resurfaces specifically during the Parthian and Sassanid periods, showing up again at sites like Lyresangbon and Vestamin in the north. The exact same style, appearing again after 2 millennia of silence. A funerary practice that outlives multiple empires, disappears, and then reappears almost intact isn't just an archaeological curiosity. It functions as a marker of cultural continuity, and potentially genetic continuity as well, tracing a thread connecting communities separated by thousands of years. By the time those Parthian tribes were digging catacomb graves again, an actual empire had already risen and fallen on top of all this ancestry. The Achaemenid Empire's core territories, under the same genetic geography framing used throughout this research, included the Iranian plateau itself, the South Caucasus, South Central Asia, and portions of modern Afghanistan. This was the empire most people picture when they hear the word Persian: vast, wealthy, stretching across multiple continents. The 2025 continuity study specifically targeted this period as part of its core sampling window, beginning at 355 BCE. Achaemenid period genomes recovered for the research come from northern Iranian sites positioned along that same Silk Road corridor running south of the Caspian Sea, the same geographic thread connecting every sample discussed so far. Genetically, these individuals sit as intermediates on an east-west genetic cline stretching across the entire Persian plateau, neither fully western nor fully eastern in their ancestry, but positioned somewhere along that gradient. What's striking is the mismatch between political scale and genetic scale. The Achaemenid Empire's reach stretched from Egypt to the Indus River, one of the largest empires the ancient world had seen. And yet that enormous territorial reach didn't translate into a dramatically different gene pool at these specific sampled sites. Political scale and genetic scale, in this data, simply don't move together. The empires changed names 3 more times before the plateau's next real genetic surprise showed up. After Alexander's conquest swept through the region, The Seleucid period briefly took control of the same territory the 2025 study sampled. Parthian rule followed next, and after that came the Sassanid Empire, carrying the study's core window forward until it closes around 460 CE. Four distinct political systems, one after another, all governing the exact same stretch of land. To track genetic change across that entire span, researchers specifically selected 3 historical period sites in northern Iran. These weren't random excavations. They were chosen because together they cover the full run from Seleucid through Sassanid, giving the study a continuous genetic record across 4 changes in imperial control. The material itself wasn't easy to work with. Genomic coverage on these historical individuals ranged from 0.20x up to 1.11x, averaging around 0.61x overall. From each sample, researchers recovered somewhere between 109,000 and 741,000 SNPs, measured against the standard ancient DNA reference panel used across the field. Despite 4 separate empires rising and falling across this exact timeline, the researchers still described the underlying population as fundamentally stable. Not identical, not frozen in place, but stable, in a way that runs directly against the assumption that political conquest reshapes a population's genetics. There's a genuine surprise buried in this same dataset. None of the sampled Achaemenid, Parthian, or Sassanid individuals carry the R1A haplogroup. Not one. That's a notable absence, considering R1A runs at close to 15% in modern Iranian populations today. That gap between the ancient samples and modern Iran only makes sense once you look at where R1A actually concentrates. 938. That's how many Iranian men Grugni and colleagues tested for their 2012 study, published in PLOS One, drawing samples from 15 distinct ethnic groups spread across 14 provinces. Each man's DNA was screened against 84 Y chromosome biallelic markers, along with 10 STR markers, giving researchers an unusually detailed picture of the country's paternal genetic landscape. Across nearly every group tested, one signal dominated: J2A subclades, described in the research as an autochthonous background, meaning genuinely rooted in the region itself, but far from uniform. The frequency of J2A shifted noticeably depending on which group or province researchers were looking at. G2A-P15 told its own story of regional variation. Overall, it appeared in 9.1% of the total sample, but that average masked enormous swings underneath it. The marker was completely absent, at 0%, among the Sistan-Baluchistan population, while reaching as high as 19.3% among Khuzestan Arabs sampled in the same study. More than a decade later, a 2024 follow-up assessment expanded the picture considerably, covering 18,184 individuals. That larger dataset found J2, including the JL26 branch, sitting at 35.64%, the single highest Y DNA frequency recorded anywhere in the study. R1A came in well behind it, at 14.68% overall, landing in second place rather than first. The real story isn't the overall number. It's where that number splits by group. Look closely at the Grugni dataset, and a clear pattern emerges. An inverse relationship between J2A and R1A running across Iranian-speaking populations. R1A climbs higher, specifically among Eastern Iranian-speaking groups. J2A, by contrast, runs higher, specifically among Persians. The numbers make that split concrete. In some Persian-speaking samples, R1A1A M198 frequencies were recorded at roughly 3.5%, a small fraction of the population. In other provinces, that same marker climbed past 20%, a difference of nearly 6-fold within the same country. This isn't random noise in the data. The pattern tracks a real historical divide across the plateau. Western populations pull more heavily from the older Zagros farmer layer, the same ancestry traced back through the Neolithic genomes discussed earlier in this story. Eastern populations, by contrast, carry more of the steppe-linked signal that moved south through Turan during the Bronze Age. Two directions, two different genetic histories, both converging inside the same modern national population. The mitochondrial side of the story runs on an entirely different set of numbers. 24. That's how many distinct mtDNA superhaplogroups the 2024 large-scale assessment identified across Iran, covering the maternal side of the country's genetic record in far greater detail than earlier surveys managed. Haplogroup U leads the list at 20.73%, with H following close behind at 18.84%. Further down, J sits at 12.10%, HV at 9.22%, and T at 8.98%. Add those 5 West Eurasian lineages together, and they account for 69.70% of all sampled Iranian mtDNA, more than 2/3 of the entire maternal genetic record concentrated in just 5 haplogroups. Within that mix, 2 subclades stand out specifically. J1 alone reaches 11.24%, and U7 reaches 7.30%, making them the 2 most common subclades identified anywhere in the dataset. The same 2024 study cataloged 14 distinct Y-DNA haplogroups running alongside this maternal picture, with J, R, G, T, and Q emerging as the 5 predominant paternal lines across the country. All of that diversity sits on top of one community that deliberately refused to blend into it. In 2017, a genetic study of Zoroastrianism sampled 526 men across India, Iran, the United Arab Emirates, and the United Kingdom, tracing the paternal and maternal lines of one of the world's oldest continuous religious communities. What the researchers found among the Iranian Zoroastrians specifically was striking. No detectable recent admixture events anywhere in the dataset. Researchers attribute that finding to a combination of founder effects, population bottlenecks, and long-term endogamy, generations of marrying within the same small community, rather than mixing with surrounding populations. When the analysis was adjusted to correct for this isolation, the genetic gap between Zoroastrians and other Iranians shrank substantially, confirming that the isolation itself, not some separate ancestral origin, was driving the difference. The dating on this admixture tells its own story. Whatever mixing did occur in Iranian Zoroastrian ancestry happened sometime between 570 BCE and 746 CE, a window older than any other sampled Iranian group in the entire study. One interpretation ties that specific window to Cypriot allies who joined Alexander the Great's conquest of the Persian Empire in 332 BCE, a single historical contact point that may have left its mark centuries before any other group's admixture events occurred. That same isolated bloodline didn't stay in Iran. Part of it sailed east. 7th century CE, and the Zoroastrian Persian Empire collapses. In the aftermath, Zoroastrians from the province of Pars made a choice that would define their community for the next 1,300 years. They fled, sailing east toward India rather than remaining under new rule. Their descendants there carry a name taken directly from their homeland, the Parsees. Genetic evidence puts the timing of their integration into India at a specific window. Admixture with local Indian populations dates to somewhere between 690 and 1390 CE, a span covering roughly 7 centuries of gradual mixing rather than a single sudden event. The shape of that mixing is just as telling as its timing. The genetic evidence points to a migration that was overwhelmingly male line, Persian fathers pairing with local Indian mothers, generation after generation, rather than entire family units relocating together. That pattern left a distinct signature in the DNA, one researchers can still read out centuries later. Today, the Parsi community in India numbers under 52,000 people, according to the 2011 Indian census. A small population by any measure, especially set against the empire their ancestors once fled. Within that small community, one subgroup stands out even further. Parsi priests specifically recorded the lowest gene diversity values of any population sample studied in this research, for both Y-DNA and mitochondrial DNA. A community already narrow in its genetic base, narrowed further still by the priesthood's own marriage practices. 13 centuries later, a single mitochondrial genome from that community tells the rest of the story. Researchers generated the first complete Zoroastrian Parsi mitochondrial reference genome ever assembled, now cataloged under the GenBank identifier MT506314. It belongs to haplogroup HV2A, a lineage that traces directly back through the Parsi community's maternal line. That single reference genome became a foundation for further work. The research team used it to build haplogroup-specific reference genomes for 100 Parsi individuals, giving them a detailed genetic map of a community that had, until this point, been remarkably understudied at this level of resolution. One detail from Parsi cultural life adds an unusual layer to the genetic picture. Parsi tradition bans smoking outright, a practice tied directly to fire's sacred status in Zoroastrian belief. It's a rare case where religious doctrine may be shaping population health data alongside genetics, a cultural rule potentially interacting with biological outcomes in ways researchers are only beginning to untangle. What stands out most, though, is simple persistence. This mitochondrial line has remained largely intact for over 1,300 years, passed down through a community that consistently married within itself rather than outward. That same endogamy cuts 2 ways. It preserved the genome, keeping this specific maternal lineage traceable across 13 centuries with remarkable clarity. It also concentrated whatever genetic vulnerabilities came bundled with the original founding population, locking both the strengths and the weaknesses of that small founding group into every generation that followed. Back on the plateau itself, the mechanism working underneath all of this had a name too. The 2025 Continuity Study's core argument is direct. Empire-level political change and population-level genetic change are two separate processes running on entirely different timelines. One can shift rapidly, a new ruler, a new capital, a new currency. The other moves far slower, resistant to disruption in ways political history rarely accounts for. Geography did much of that work. The Zagros Mountains, combined with the plateau's internal basins, physically limited how thoroughly outside populations could ever fully replace the people already living there. Mountains and basins aren't just scenery in this story, they're barriers that shaped who mixed with whom, and how much. Trade brought contact without triggering wholesale replacement. The Silk Road connections running south of the Caspian moved goods, ideas, and some people through the region constantly, yet that flow of contact never translated into the kind of population turnover a casual observer might expect from centuries of open trade routes. Endogamous practices, visible clearly in the Zoroastrian data covered earlier, show one specific mechanism by which certain communities preserve their ancestry across repeated changes in ruling power. Marrying within the group, generation after generation, insulated specific lineages from the churn happening around them politically. Even the larger steppe migration fits this same pattern. Steppe ancestry entering through Turan mixed into eastern populations on the plateau without erasing the older Zagros farmer signal still present in the west. Two distinct ancestral layers, sitting side by side rather than one replacing the other. The result across all of this is a layered gene pool, added to repeatedly across thousands of years, but rarely erased outright. The 3,000 years of continuity in this story's headline is really a story about resistance to loss, not an absence of contact. Only 2 medieval genomes exist from the entire Iranian plateau, according to the 2025 study. A strikingly small number for a region this thoroughly documented in written history, temple records, and imperial archives. The gaps don't stop there. No confirmed royal Achaemenid or Sassanid genome has yet been published from a verified elite burial anywhere on the plateau. For all the written history surrounding figures like Cyrus, Darius, and the Sassanid kings, there's still no genetic sample confirmed to belong to any of them specifically. The material researchers do have remains thin by modern genomic standards. Coverage on the historical samples analyzed so far averages around 0.61x, enough to draw real conclusions. But far from the deep, high-resolution genomes researchers can now generate from more recent remains. Researchers themselves are candid about the scale of what's missing. This dataset covers only 9 sites, drawn from a plateau that spans multiple modern countries and thousands of years of continuous habitation. 9 sites is a start, not a survey. And yet, every new sample analyzed so far has added something specific to the record. The early Chalcolithic outlier, bridging Neolithic and historical genomes, the Shar-i-Sokhta migrants pointing toward the Indus Valley, the conspicuous absence of R1a among Achaemenid, Parthian, and Sassanid individuals, despite its presence in modern Iran. The next batch of samples could just as easily overturn parts of what this episode just covered. That's the part of ancient DNA research nobody puts in the headline. The story keeps being rewritten one skeleton at a time. 3 named empires sat on top of this population. 4, if you count the brief Seleucid interlude between Alexander's conquest and Parthian rule. Underneath all of them, the population itself mostly stayed the same. The Zagros farmers of 8000 BCE left a genetic signature still measurable today, most clearly in Zoroastrian communities whose isolation preserved that ancient signal better than almost any other group tested. Steppe ancestry moving south through Turan, Indus Valley outliers turning up at Shar-i-Sokhta, and that older Zagros ancestry all sit layered together in the modern Iranian gene pool, one on top of another, rather than any single wave replacing what came before it. Endogamy preserved specific threads almost perfectly. The Zoroastrian line in Iran and the Parsee line in India both carried their ancestry forward nearly intact for well over 1,000 years, protected by the same marriage practices that kept outside genetics from ever fully entering. Every new archaeological site sampled across this research has added a name, a date, and a number to a story that is still very much being assembled, piece by piece, skeleton by skeleton. If the throne changed hands 9 times across 3,000 years, and the people living underneath that throne barely moved at all, it raises a real question about what actually makes a nation the same nation across that much time. The rulers sitting on top of it, or the ancestry running underneath? That's a question no lab result can fully answer. Only the next dig can add to it.